And on behalf of all of us at the Explorers Club, welcome. My name is Ted Giannoulis. I was president 2 presidents ago, but delighted to be here introducing tonight's talk. I'm especially excited because I recognize almost none of you. I love that. I thought Martin, that was kind of it. So just out of curiosity, how many people are here for the very first time? Yeah, that's good. So if you haven't taken a tour, I highly encourage you to. I'll just spend 1 minute for those who are new, just because it's to give you context of the magical space you're in. When you walked inside this Harry Potterish building, you saw a plaque that said the 5 famous firsts. And that's kind of the core and the history of the club. First to the North Pole, first to the South Pole, first to the top of Everest. Sir Edmund Hillary was our honorary chair before he passed. First to the bottom of the ocean, Don Walsh, and everyone since then, whether it's James Cameron or more recently Victor Vescovo, all members of the Explorers Club, and then first to the moon. And in fact, you may have seen a tiny little flag in there that was the flag, one of the flags from Apollo 11. All the lunar landings carried Explorers Club flags. My favorite thing in the entire club is not here right now, but usually the astronauts carried flags were about this big and they'd stuff them in their sleeves. But for some reason, there was one lunar landing that decided to carry a large flag, this size flag, and that was Apollo 13. So for those of you who've seen it, you know how it kind of ended, not particularly well, but safely and a happy ending, thankfully. But we have the original flag in the baggie, unwrapped, with a note from Commander Lovell saying, "Sorry, didn't get a chance to unpack your flag." Kind of busy going into hyperspace, and a note of apology. It's just beautiful. And the phrase I do remember, you'll have to come back when it's back and mounted, his description of Apollo 13 was, "Plans were disrupted." And that was the Apollo astronauts. So Apollo astronauts are very cool, but there's all kinds of other cool explorers of the club because it really grew from those roots. And I do have to say, before we move off what we call the famous first, you're actually here for famous first tonight, which is Felix Freeland, who's hiding in the back now that I've said this, is now on behalf of the club chairing and running the whole Public Lectures Committee. And this is his first one ever. The record, Felix, is 7,912. So good luck with us. But to give you a sense of the club today, we have all those different disciplines have grown and spread. We have 3,800 members all over the globe now, and they do everything. It's places like the first time to outer space or deep sea or caves, but also it's about going back to places you've been with a new set of eyes, new technology, new sensibilities. People care a lot more about conservation, preservation, et cetera. All those things are kind of coming together. And so today, the Explorers Club runs all kinds of programs, one of them being public lectures, but we also have Climate Week and Ocean Week and all kinds of educational learning opportunities. We provide grants for a number of different organizations. Rolex has been a sponsor for many, many years. They sponsor grants out in the field. So we're really trying to be more inclusive in our efforts to get more people out exploring, but also a real focus on the next generation so that we're not resting on the laurels of those wonderful people that are referenced in the lobby, but really expanding from there. And the other thing I would mention, the reason you have to take a tour is the other way of looking at this place is this remarkable building that was built in the early 1900s. And it would be too much to go into it now, but it's a very wealthy family, and all kinds of things around it are significant from that age. We're huge art collectors. There's all kinds of details. And if you take a really good tour of the club, you'll get a sense of that. So please take us up on that. But the room that you're in right here, you've got these flags that have done so many cool things that they get retired, so don't get lost, stolen, or broken. This is the Kon-Tiki flag as an example that Tor Heyerdahl took. That is one of Peary's sledges from the trip to the North Pole. So you're in a very magical place where people tell very magical stories. And at the heart of the Explorers Club is curiosity about the world around us, but also then taking that curiosity and doing something, converting it into actual action and doing something with the knowledge that you accumulate over time. So with that in mind, we have a very bold presentation tonight, which is probably why some of you are here that haven't been here before. So welcome again to all of you. But Peter and Leonid, you've seen their descriptions and their bios. I was able to spend a little time with them beforehand, and it's fascinating. You could look at it kind of like sailor meets scientist, because they both have these incredible, strong roots. So Lana in science and Peter in oceans. But when you see them together, it's really an organic thing. And they really come across, along with their other team members who are here tonight, as a team on a mission. And the reason, one of the reasons why the biodiversity and genomic projects are so interesting, just to give you a couple of glimpses, 3 in particular. The first one is that if you think about climate as a huge gating issue of the day, more recently, over the last couple of years, oceans has really moved up. But now with the biodiversity treaty and a sensibility around the importance of biodiversity, biodiversity has really come to the fore. And if you're going to do anything around biodiversity, like create credits or measure marine protected areas or figure out if sinking seaweed is going to be helpful or putting olivine in the ocean, to do all those things, you have to start with baseline. So baseline is a word that you hear more and more these days, and that's what one of the things that these folks are creating. The other thing I think is fascinating is if you look at the history of ocean research, and there's some great books written on it, I'm happy to tell you about them later, but if you look at the history, a lot of it is physical oceanography. And there's been not so much, there's been of course a lot, but compared to the physical oceanography and chemistry, biology is still in its up and coming phases, and these folks are right in the middle of all that. And the third thing about it is it's got a little bit of a feel of a rocking adventure to it. I mean, you're thinking really big, you're building boats, you're pulling people together, you're putting this great science together, and you're moving forward at a rapid speed for all the right reasons. And that kind of spirit of adventure, but also meaning and bringing it back and communicating is what we're all about. So without further ado, I'm going to introduce our speakers today. Thank you so much for joining us, and I hope you'll come back more. Thanks. So I'm going to leave that.
Well, it's a pleasure and an honor to be here at the Explorers Club and to share our work. Good evening. So I don't know how many of you have gotten an encouraging call from a Nobel laureate. I haven't, but Dr. Leonid Moroz did. Christmas Eve 2002 in his laboratory at the University of Florida. Now, Dr. Moroz is a distinguished professor of, wait for it, neuroscience, genetics, chemistry, and biology at the University of Florida College of Medicine. At the time, he was an associate professor who had been recruited from his work in Antarctica. Oh, sorry, I'm looking at the wrong ones. In Antarctica, where he was diving under the ice to analyze the blood and the proteins in the fish so that they're not freezing at minus 1.8 Celsius. Dr. Moroz came to the University of Florida to study neuroscience, the origins and architectures of the original neurological systems on this planet. At the time, people were talking about neurotransmission. They were talking about pathways, neurotransmitters, certain biochemicals. But Dr. Moroz had a hunch that there was something going on at the DNA level, cell by cell by cell. And he started to use some of the new techniques that were developed in the Human Genome Project, shotgun sequencing in a word, to analyze different neurons. It was not going well. His department heads were not encouraging. In fact, one of them said he would never get tenure. His colleagues were tepid at best. But he got a phone call. That phone call I mentioned in 2002. He expected it was his wife, Tatiana, because it was Christmas Eve and it was probably time to get going home. And he was slightly surprised to hear the Germanic accent of Dr. Eric Kandel. Dr. Kandel said, Dr. Moroz, because yes, because I've seen your presentation to the Society of Neuroscience, and I think you're onto something. To give you some background, Dr. Kandel had won the Nobel Prize 2 years previously for describing the cellular and molecular basis for learning and memory. On top of that, for Leonid personally, Dr. Kandel had written a book called The Cellular Basis of Memory that Leonid read as age 19 as an undergraduate at the Belarusian State University in Minsk. And it actually kind of set his scientific, he would say that it set his scientific path forward from that time. So Dr. Kandel had basically described, and even today, 50% of what we know about memory comes from the understanding of Aplysia californica. It is the sea hare. It is a beautiful organism in the Aplysia family. It's found, this one, around the coast of California, and it has enormous neurons. You can see the neurons by your naked eye. So we've learned a lot from that. So it was much more than an encouraging phone call, because Dr. Kandel said, Dr. Moroz, I would like you to collaborate with my laboratory at Columbia. And in fact, they collaborated for 20 years until Dr. Kandel retired last year at the age of 93. So armed with this encouragement, Leonid continued to look at marine organisms. And he was gathering them around the world, and he was running up against something really, really problematic, which is they all degraded by the time they got to the lab. They were mainly gelatinous, they were mainly fragile, and he was not getting the genomic reads off of them that he would like to. So he said, it's time to go to sea. He assembled a laboratory in the back of this, behind him in a container, and he put it onto a 141-foot vessel called copacetic. It had two, good name, it had 2,000 horsepower engines, a crew of 15. And they went across the Bahamas between the Keys and the Bahamas, and basically were able to, for the very first time, sequence an organism at sea. Dr. Moroz sometimes calls this the genetic equivalent of going to the stars. You have to understand that before this, these kinds of labs were really restricted to the largest institutions in the country. And the idea of putting it all in a container and putting it onto a boat took a certain turn of mind that, as it happens, Dr. Moroz has. And it was a breakthrough trip. They were able to sequence a number of different organisms. But interestingly, Dr. Moroz's research kept coming back to the ctenophore, which is also known as the comb jelly. At the time, it was considered essentially kind of a runt jellyfish. Nothing really interesting, nothing really noteworthy. But when they started looking at the genomic reads, there was something really remarkable about this. It shared very little in terms of neurotransmission and architecture with any of the other sophisticated marine organisms. And with his previous research and the reads on this voyage, Dr. Moroz wrote a paper in Nature where he basically upended 150 years of evolutionary history. And he said that this organism branched first from the animal tree of life and not sponges. Now, sponges are pretty simple, as you guys may know, but they actually have a neurosystem. The question was, how could you have something that branched first that became as sophisticated as the ctenophore? It's a revolutionary idea, and it has, in fact, opened up a wide range of different applications, which we'll talk about later in the second half of tonight. Fast forward to 2018. We are off the south coast of Molokai. This is Dr. Moroz's very first sailing trip. Previous to this, a year before, we had gone with one of his PhD students from Cabo San Lucas all the way over to Hawaii. We were put together by a group called Seakeepers. Dr. Moroz was so convinced that it was going to work that he didn't go himself. He sent his PhD student. But a year later, given the results, he thought he'd come along. So basically, we spent a week doing a couple of dozen plankton hauls. Plankton hauls are essentially large windsocks put into the ocean at different depths with different microns. And Leonid would spend hour after hour looking into his microscope. In fact, the crew really didn't know what he was looking at for that long, but this gives you an example. Absolutely remarkable creatures from every single haul that we did. And it was a really important and interesting week for everyone on board. At the very end of the week, this is the afternoon we had this conversation. You can see it's a pretty simple operation. The line is in the water. We leave the plankton haul in there for 15 to 30 minutes. And Leonid and I are waiting for the plankton haul to come up. And I said, Leonid, this has been an incredible week. It was whale season, so the whales were jumping. We saw rainbows. I'm like, we'll do this as long as you want. But does this really help science? And I won't forget when Leonid turned to me and he said, the biggest impediment to understanding the ocean is the cost and the complexity of the research vessels, which was not the answer I was expecting, to be honest, and a conversation I've been waiting to have for a long time. He went on to say that most research vessels are incredibly capable, but they're expensive to build, they're expensive to operate, they vibrate all the time because you have machinery and generators on board all the time. Very difficult to do biological work in that way, especially under microscope. You have to pack multiple teams on board to cover the costs, and you have a certain amount of time because these things are big and they have to be resupplied with fuel and food, et cetera, and so nobody gets the chance to do the work they want at the time they want, the season they want, for the organism they want, and it doesn't work, is the short answer. And I thought to myself, this is a problem we can solve. This is the vessel SAM. It is a 1990s 48-foot aluminum, all latitudes ocean expedition vessel. My family and I had the opportunity to buy it 5 years previously. I was born in San Francisco, and my parents brought me out for my first sailing trip when I was 3 months old on a 26-foot sailboat. I went on to have a little dinghy of my own and race in high school and college. And San Francisco is notoriously windy, cold, foggy, with lots of currents, and I loved it. So I went on to become an expedition leader for the National Outdoor Leadership School, doing sailing and sea kayaks in Baja and British Columbia, and also doing some expeditions in Patagonia. I had a chance to sail the Flyer, which was the Whitbread winning around the world boat from Cape Horn down to South Georgia, Shackleton's Grave, up to Buenos Aires. And I kept thinking to myself, this is an amazing tool, a vessel, an amazing tool, a sailing vessel. How and why can we put it towards real scientific use? So that was really the beginning of our collaboration. And for the last 5 years, we've covered close to 15,000 nautical miles across the whole North Pacific and all the way across the Atlantic. And we've seen some things. This is crossing the Shelikof Strait between Kodiak Island and the Alaska mainland. We went from Hawaii all the way up to the Aleutians, the whole Gulf of Alaska. And we found incredible organisms. Every picture you see tonight is from our voyages. They are fascinating. The work is relatively straightforward. You have some arm building exercise pulling these things up. And again, hour after hour of observation on these amazingly powerful microscopes. And these are some of the images. Absolutely remarkable. Leonid calls these the aliens of the sea. And we went to some really remote places. Here we are sailing along the Wrangell St. Elias Range. This is the largest viewscape in the world. You can see from the sea level to 18,000 something all the way up to the top. Again, sea angels on the side. On the right is one of our ancestors. It has a chordate, which is a backbone. We went along the British Columbia coast, western Vancouver Island, and again, saw wonderful organisms. We were invited by the Vincent and Antarctica folks to cross the Atlantic from Holland to Uruguay. We raced across, and then we found these are sea angels, equatorial sea angels, important for understanding mood regulation and muscle tone or muscle use. We went across the Sargasso Sea. We found completely intact coral larvae in the middle of the equatorial sea, which was a big surprise. Sometimes we do not have fancy equipment. Leonid, Matt Stromberg's here, who's our crew member. Leonid saw a jellyfish he's been looking for a decade, and Matt ran to garbage can and found a red Solo cup, and he was able to get a dozen of them. So we use what we can. But the whole crew has learned how to do a lot of the work, and we've trained a number of people. We've retrofitted a van and went to very remote western Vancouver Island and used their trailerable fishing boat. And from a skeptic 5 years before, I took this video from crossing from Kodiak Island to Homer. So Leonid, we're here just past the Barren Islands on our way to Homer. Tell us about the last sample we just took.
Surprisingly, it's one of the richest samples we ever collected. We got at least 120 macroanimals in this sample, including weird ones like endoprocs, several ctenophores, larvae, jellyfishes, probably 5 or 6 ones. It's incredible, and no much algae, no much toxicology, so it's unbelievably clean water. I think it's a jackpot.
Excellent. On our way to Homer.
Yeah,
what a week.
Yeah.
What a week.
Fantastic.
So we went from a skeptical scientist to jackpot in 5 short years. This is off the coast of Maine last summer.
Yeah, what do you got there?
Yeah, it has kind of a lobster larva. I'll photograph this lobster. Yeah, but it's really tiny. Very colorful.
So at this point in the evening, you may be asking yourself, what is a neurobiologist learning at sea? Obviously, the pictures are amazing. The diversity is amazing. To explain, I'm going to use one of the most common, powerful models known throughout the world, and that's Lego. On the left, you can see that Lego makes a Death Star that's got 3,400 and some odd pieces. Impossible to put together without the instruction manual. Still very painful when you step on the pieces. On the right, for anyone who has kids, on the right is the human brain with 86 billion neurons. The human brain in its current complexity is impossible to plumb. So how do we figure out how our brains work, our fundamental neurobiology, not just our neurobiology, but many organisms? We go back in time to 100 million years ago. This is the organism that Dr. Eric Kandel studied for as many years. Lego from, let's call it the early 1980s. You probably could figure it out without the instruction manual. What we're really doing is going back another 450 million years to find the original pieces of Lego. The original pieces of how biology evolved on our planet. And ctenophores, as it turns out, which I talked about earlier, are in fact one of the original Lego pieces. How many pieces of Lego are we talking about? These are pictures from the MODIS 2 satellite that was put up by NASA in 2016. All these beautiful blue swirls that you see here off the coast of South Africa, green and blue, are all plankton. There are 321 million square miles of ocean, cubic miles of ocean, rather. They are blooming, dying, living all around us. Trillions upon trillions of organisms that are really the hidden beating biological pulse of our planet. They are being moved around the oceans with wind and waves every day, every sunrise, every sunset, and have been doing so for hundreds of millions of years. And we really know very, very little about them. Plankton is important for all the air 60% of the air we breathe, carbon capture, it's the base of the food web. Really, it is ultimately the core of the web of life. This is a picture of one drop of ocean water that we collected in British Columbia. And every single one of these organisms is a genomic and evolutionary miracle. Otherwise, it wouldn't be in this picture. Right? It is, each one of these organisms represents experiments that have been conducted by nature for 3.5 million years. How can we figure out what the secrets are and understanding planetary health, fundamental biology, and human health? The challenge is that all of life began in the oceans. We do not understand them, and as we all know, we are losing these organisms. As we know, presently, maybe 100 species a day. So what our charge is to figure out functional biodiversity. By that we mean the dynamic of traits of cells and species. What essentially we're trying to figure out is every organism has homeostasis, which keeps itself alive, but then it also creates a healthy ecosystem by connecting with other organisms around it on the genomic level. And we have to go out there to find it. And if we look at it, we maybe know 10% of the marine species, the big ones, maybe. Small microorganisms, maybe less than 1%. Functional marine biology, how they communicate with each other on genomic level, less than 1/10 of 1%. And here's the trick. You take one of these things out of the ocean, they'll have 1,000 gene changes in 15 minutes. So if you really want to understand how the world works, you have to go out and you have to get them fresh. So our work is to do extensive sampling with top scientists on flexible, low-cost platforms, getting high-resolution genomic data, training lots of people in the methodology, and making it all open source. It's a tall order. The impact, we believe, is multifold. Biodiversity baselining, as Ted mentioned earlier, planetary health, ocean trend tracking. Again, we talked about fundamental biology and evolutionary, what Dr. Morrow's early work was on the brain. Biologically active molecules, 70% of all the drugs that we have are based on, are nature-based, like aspirin from birch bark. And again, to make it open source, so even scientists who are not on the vessels can have access to it. These are the costs and the problems right now. Technology has been too bulky. Much of biological science has been basically comparative biology, which is essentially what Darwin did. Cost, the vessels are too big to build, operate, and time. As I mentioned, the trips were short in length. So our solutions are science-based. We're essentially taking advantage of 20 years of genomic science development. Single cells in 2006, real-time seeing the three-dimensional structure of DNA came around 2018. A lot of the machine learning to actually analyze all this genomic data came in 2020. Biodiversity coordinates is essentially trying to map ecosystems in 2022, and our job is to put it all on a boat in 2024. I want to talk for a second why we deal and work on the single cell. Every genome, every cell in your body has a different genomic, it has the same DNA, but that DNA expresses differently. And those cells build up to become tissues, those tissues become organs, those organs are organized by a neurosystem to become an animal, an organism, and that organism goes out in the world and reacts to it. And signals come in from nature, and again cascade through that same chain back down to the single cell. So if you really truly want to understand how an organism lives, survives, eats, reproduces, repairs itself, et cetera, you have to be at the single cell level. So everything we do is based on 4D plus genomics. There's not enough time to go into metabolomics tonight, but I'm going to give it a quick shot. 2D, ATCG, we've all heard those letters in our DNA. DNA folds in the upper right corner, looks like a bowl of spaghetti, but essentially the way DNA folds is where the genes switch on and off. We look at the 4D as watching this observe over time. So we bring an organism on board for multiple days and we watch it and we take cell cultures. We do RNA single cell transcriptome, proteomics, metabolomics, and lipidomics. To talk about the scope of the work in a really small scale, I want to go big, and the Andromeda Galaxy comes to mind. This is a picture in 1888. This was the technology, first known picture of the Andromeda Galaxy. This is a picture of a star being born from Hubble. This is the Large Magellanic Cloud. About the same distance, actually, 163,000 light years away. Last year, the ORSIS project actually landed on an asteroid, picked some of the dirt off, the rock, and landed in the desert in the west. We're trying to do this at the cellular level. Not just observe an whole organism, but go deep inside the cell and get really pertinent information. So our laboratory on board is based on this. First, we have to get the organism fresh. And we often talk about this as sushi-grade plankton. Right? Because as I mentioned, they start to degrade and change very quickly. So we want to do an observation, not an autopsy. Second, they should stay alive for ideally 3 to 4 or 5 days. We do a series of physiological observations and tests. And we do very high resolution photography and videomics. We get ready for multi-omic sequencing off the ship at this point, and then we integrate it into a larger database later on. We do this first and foremost by putting together state-of-the-art labs. Currently, we've put them onto these mid-sized aluminum vessels. As I mentioned, we've retrofitted a lab van, and we are designing new build vessels for the future with Dijkstra Architects in Holland. And again, I want to emphasize that all the data goes to open source. We've been recognized by the UN as part of the Ocean Decade as an ocean shot project, much like a moonshot, combining two disparate threads of vessels of this kind with this kind of ocean data. And I just want to go through like life on board and practical applications. So these are our routes in the last 5 years, the whole North Pacific and all the way across the Atlantic. These are the vessels that we've had, ranging from 48 feet to 77 feet. Most of the work, we don't even retrofit the vessel. We're just using, we built a lab table on top of the back room, the back deck in Vincent. We use the plankton halls. We've built our own types of aquaria and sea tables, and we have the plankton nets that I mentioned. But then things get a little bit more complex. The microscopes are amazing these days. And a lot of these videos that you saw in photographs were taken on an Apple iPhone. This is probably one of the first pictures ever of a petropod mollusk releasing fertilized embryos that we know of. We've done dissections on board. This picture on the right, this is an antecedent to the aplesia that I mentioned, that Dr. Kandel mentioned. What you see on the iPhone right there is a full functioning neuron. Not for long, sadly, for this organism, but for science. On the left, this box that looks like a home printer is a fluidics library maker. It can disassociate 100,000 cells a day. It can puncture that cell and barcode all the RNA in that cell. It replaces 1 million pipetting motions in the laboratory. Costs about $150,000, so we're careful with it, but it works at sea. On the right, the kind of fridges you imagine, but I'd just like to point out that the one here, the Sterling, was developed for delivering vaccines during the pandemic. It can hold minus 80 Celsius at 4 amps. We plugged it in Holland, we unplugged it in Uruguay, it worked the whole time. So we can keep samples at scientific standard when it used to be a box like twice your home fridge that weighed 600 pounds. We now have 3 of them. So Starlink. Last year we put this on board. Besides the fact, we got a Gumby Award once for being the most flexible crew helping scientists on a difficult launch of their equipment or landing on an island. And Leonid had never seen Gumby, so the first thing we looked at when we put up Starlink in the middle of nowhere was a Pokey and Gumby skit from the 1960s, which for a Russian scientist was really surprising. But nonetheless, we could do 3 Zoom calls at a time on this, which keeps scientists abreast of their work back at home. But most interestingly for us, we were able to upload the videography the night before, and when we were making coffee the next morning, we already had a full artificial intelligence read from a colleague at the University of Washington who'd done a physiological read of the whole organism. Why is this important? Besides the fact it's super cool, it actually tells us, like, is this an organism that we found before or not? Should we focus in this area? Should we move on? So some things to think about. Western Vancouver Island, we'll talk about this. We were able to go out there for $500 a day. We set up a lab on board. And we'll talk a little bit more about that later. We're so convinced of this whole method that we've purchased a 65-foot aluminum vessel, a canter. It's in Holland. We are currently rebuilding it to do all of these next level from what we've done on the vessel Sam and Vincent before. It cost us $650,000 to purchase, and we're raising $500,000 to rebuild it currently. We gutted the back. We've done laser scanning inside and outside, and we've basically designed a whole upstairs gathering with plankton, trawls, and also observation area for physiological tests and videography. Down below will be the 2 genomic stations, including sequencing, and to our knowledge, this will be the first-in-class vessel in the world that are able to have this capability. And we are going to go around the world. Our idea is to essentially follow Darwin's route. There's a lot to learn, and I want to just point out something. You guys see these dotted lines here? Those are the most studied parts in the world. Not surprising, right? West coast of California, Northeast, United Europe, industrialized, wealthy countries, the least diverse parts of the ocean. Much of the world, especially the Coral Triangle here, which may be the most diverse point in the Earth's history, has largely not been studied, and especially not in this manner. So we're going to get out there and we're going to do a lot of different things. So one of them is to train scientists and crew. Those who've been on scientific expeditions know that morale, communication, et cetera, is incredibly important. We're going to do biological routing. So most of the RVs these days, research vessels, have a start date, an end date, they know a port. You don't leave fish to find fish. right? So if you get into a hotspot, we have to figure out a way to maintain the scientists, the supplies, the equipment, and the vessel for biological routing, continue to develop sampling and laboratory protocols. We work with a number of institutions around the world, including the European Molecular Biology Laboratory and places in Japan, Australia as well. And we're going to work on regional training workshops. And I want to focus on this a lot. We are going to spend half our time training people in remote locales around the world. We will never be able to build enough ships to truly map the oceans. We need to get people who live in their own ecosystem, understand it for hundreds, if not thousands of years, and train them on a range of skills, from crews to fishers to scientists. Areas that have not been traditionally resourced enough to have major marine institutions. So if Woods Hole and Scripps and Friday Harbor were the breakthroughs of the 20th centuries, small regional mobile labs and vessels will be the research stations of the 21st century. Collaboration will be key to really expanding our work and continuing to work with a range of new students. We've done this already. Dr. Moros has trained over 300 students in his methodology. In 2002, these are pictures on the right from San Juan Islands, Friday Harbor Labs. We had 16 students there from advanced undergraduate to postdoc, from Sri Lanka to Italy to China to the United States. 70% of them went on to study this methodology in more detail. It's a key part. We want to wrap up all our knowledge and to build vessels. We think the throughput scientifically will be much greater than retrofitted vessels, and we've already started to design those, but you always learn more as you do it. Much more expensive, but still much less expensive than traditional research vessels. I want to wrap up this introduction by bringing a great quote from Sidney Brenner, who also won the Nobel Prize. Progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And that's really our methodology. We don't know much about the oceans. These tools are just newly available. Our charge is to get out there quickly, get out there inexpensively, but using top equipment with top scientists, and really get a snapshot of the health of our planet. And it's pretty fun too. And intellectually fun. So with that, I'd like to invite Dr. Moros up to talk about the implications of a number of 3 different voyages we took. We narrowed it down to 3, and to delve into some of the scientific implications of the work. Leonid, come on up. So, Leonid,
Just would like to say thank you, because we have to be kids, right? So kids like to play, like to explore. And sometimes when we become too smart, we start to be very pragmatic. But this is what drives science and society. When you want to play, look on the rocks. Scientists sometimes do it all of the time. And I would like to say, but because technology become more friendly, like iPhones, there are more and more science citizens, which in biology do more than half of work. It's incredible websites, incredible contributions, and it's intellectually very engaging. Yeah, so.
Leonid, we've been on many voyages together, and a lot of the crew, some of who are here, we have board members here as well, and a number of them online. We have had some fascinating discussions at the end of the day in the main salon of the vessel, which is wood paneled. And we will just ask you, like, what did you see today? And about 2.5 hours later, we have been down this incredible biological journey of the implications of the secrets of the ocean. And since this room looks a little bit like a vessel, I thought we'd try to get that mood going. So you started as a neuroscientist, but today you are collaborating with mathematicians, computer programmers, artificial intelligence people, synthetic biologists. Let's say your worldview has expanded a lot with going to the ocean. So I want to talk through three specific voyages that we've been on, and to talk through some of the implications. And the first one I want to talk about is the van in Friday Harbor. So backstory, we retrofitted this van. We just spent 2 days filling with all the equipment, and we're rushing to catch the ferry from San Juan Island over to the mainland and then up to British Columbia and Vancouver Island. And Leonid gets in the front seat, and we're driving up the driveway, and you said, "Bob thinks I'm wasting 2 weeks of my life." To give you an idea, this was a very famous zoologist who's worked at Friday Harbor Labs for a long time. And I was sitting there at the driver's seat thinking, that's not really what I want to hear right now. But we did cross 2 ferries and then drove down 60 miles of dirt road. And the very first plankton trawl that we did using the fishing boat that we brought with us completely, frankly, blew your mind and changed it. Why was he wrong, and what did you see?
Well, I would say I was wrong at the very beginning because I was agree with him. Well, thinking about, I'm study brain, and he is also very famous from neuroscience standpoints. And you have a lot of stuff to do, and then somebody said, okay, let's drive another 60 mile north, and that maybe brings something else. So common sense say no. Exploration spirit says, yes, you should do it. And yes, next day I realized I was completely wrong, and he was wrong. And just to give you in perspective, Friday Harbor and that area around the Seattle, San Juan archipelago, it's one of the best studied component in the United States. Laboratory was there for nearly 100 years. It's famous in many countries with enormous contribution to a different field of biology. And when you're sitting over there and have a person who has about 30 years experience on that one, and say, no, you just probably would agree with that. And when we went, well, thanks to Peter, motivation, when we came to that place, in the very first plankton, we discovered approximately 100 species and some organisms, which was ctenophores, which should not be there by all common sense, and they present in all developmental stages. So that was really incredible. But what was implication, I would say, for planet? Because in this case, we did find nursery, like seeds, which fuel almost very specific. Because you will think that animals everywhere, they are not. They sit in specific patchy places. Well, not sit, they live. And in these specific patchy places, these maintain biodiversity, and in some way whole fishing industry. Because what is really striking and not always clearly appreciated is that biodiversity means stability in entire ecosystem. If system is not diverse, it's not stable, and then you have algal bloom, pollutions, decay, bad smell. You can see it, right? So keeping diversity means that that system would be more resilient to any stress perturbations. And even today, I don't want to criticize and be skeptical for many efforts which try to help to save a planet, control negative effect of humankind on universe. But sometimes you forget that diversity of the patchy spots, this is what keeps everything alive. And we found basically 60-mile nursery, and then we found another nursery. And then we recognized that different nursery have different type of animals. So basically, message was, yes, you have to keep diversity, but it's extremely patchy. So you need to preserve all of these potential patches. And some organisms, like Starromedusa, for 30 years traveling around the globe, I never seen it. It was a very small goose island which basically size maybe a bit 3-4 times larger than this building, right? With the sea wolves only uninhabited. And that can keep this nursery for virtually entire northwestern Pacific. And these examples can flourish. So basically, text-home message, you out of the blue discover natural field laboratory, which existed for so many times. You discover diversity of the species, very patchy, which support more than 20 phyla in this area. And actually, if something going on, they just provide seeds to recover ecosystem and health. That was basically out of mind in spite of many, many, many explorations. So first reactions for many scientists may say, don't do it, maybe cost effective is waste of time. But nature shows we are always wrong. That it basically was take-home message of that. So we continue to be wrong, and hopefully we'll correct it. Did I answer your question?
Yeah, that was in August, which is not prime plankton season. Usually it's in the spring when the spring comes and life blooms. But we went back again the next year, further up the coast in the spring, and again you were surprised.
Yes, because
you were maybe afraid the first time was a fluke.
Well, exactly. And when I just told to many of my colleagues, they said, well, you were wrong. You see how much we found out. They said, well, it happens once in the season, right? And well, never happens again. And we went to another season, and we can see even better. And what is, again, myself and I have many colleagues, it was not a single trip when we do not discover something new. And then maybe, I would not say we brainwashed, but if you go to library, you can see tons, volumes of zoological literature about diversity of the animals. And you may think about, wow, we reach saturation, just Darwin, Humboldt, just all modern science all around the world. And then when you go to this kind of Goose Island and something else, you see, well, every third species is not described in the book. And when people start to do it systematically, especially with new tools, they realize, well, we may know only about 10% or less. That was really shocking. Well, thinking about, right? So much efforts, but without advanced technologies. And you just to see 90% is unknown. It's good to know what is unknown, right, in general. But this is what actually came to us. So there's a 2 million described species, animals, and estimates right now between 10 millions and 100 millions. And these tiny creatures, they provide critical link between microbes and big organism. Without that, this whole life cycle would not exist. That was shocking. So our life, our health here in New York, in Florida, in China, anywhere, right, depends on the tiny creatures which provide link between microbes which dominate our planet and us. Without that link and diversity of this link, our planet would be not healthy, and then we know that, well, 90% needs to be explored. Look on the rock. So second
going in the
trip
van.
confirm that it should be done. Yes, exactly.
Right. So let's talk a little bit about going back in time, your first voyage on when you put together ShipSeek. So ShipSeek was the 141-foot vessel that you went across the Gulf Stream from the Bahamas. Oh, by the way, we went pretty fast on this trip. This allowed us to go see those patchy areas in real time, and this was the van with our steady crew. Now we go back to 2013, 2014. You basically crossed the Gulf Stream, which is the major current of biology that runs all the way up to the east coast of the US and over to Europe. Putting together that trip was hugely ambitious. I mean, you had to put together a whole crew. You had to get funding for a real lab on the boat. You had to get the boat. talk about the power of the technology and what you learned, for example, specifically about the TINA4 that gives you an idea. This is the lab on the outside. It was a big crew. How long were you out for?
Yeah, that was about 10 years ago.
Yeah, but how long did you go out for on those trips?
Altogether, it was about half months.
Right, 2
Right.
weeks. 2 weeks of work.
2 weeks of
And the
work.
very first time an organism was sequenced at sea. But let's talk about the TINA4. Why is an outlier
Okay.
something that's so parallel
you
evolution
ask-
so important for understanding the rest of
You
biology?
have two questions. Why ship-seek and technology? And second, what about this TINA4s? Well, for the first question, if you look all beautiful books about explorers, right? Usually you just come, take picture. At the very best, you just grab something, kill it, fix it, basically remove all life. So all great contributions of many explorers end up through this way. And so basically, for nearly, I would say, 100 years, even modern science, it was very moderate tools, but still the same logic, come and kill. But when you do this, you basically not only destroyed life, but you just destroy all these connections between cells, organs, and tissue, etc. It's critical. So you lose a lot of information, more than half. So question is, all right, ideally you should bring animals to the lab. But majority of the animals critical to planet and science, and we can talk about this in a few seconds, right, cannot be bring to the lab. So, well, my crazy idea was, well, why don't we bring advanced lab to the sea? If you cannot bring animals to the sea, let's bring lab to the sea. And at that time, genomic become more democratic. And we also were wrong in some way because we were thinking, oh, if you will get genomic blueprint of everything, you'll understand everything. But at least first step was, okay, let's bring technology over there, marry technology with exploration, and that probably will give us something new. Well, in reality, it's give us something new. I'll talk about this in a few seconds. But we also find out when we look at this different type of genome, we realize there's so many new things and so many new regulations, and that is a good example. That creature, TINA4s, Camjelly, was considered like some kind of neglected medusa-like creature. Who cares? It's so fragile. You'll never bring it to the lab, or you can bring only one or two, some of them critical for fishery, but majority of this really neglected. So what is the all fun about it? And when we start to get genomic, and also we managed to do physiological data and microchemical data and neuroscience and behavioral data, I would say two textbooks were changed. First, it was quite surprised. We discovered these guys have completely different neurosystems with completely different language of neurosystem. It's basically like real aliens. So nothing which was, with few exceptions, which was in textbook about neurosystems, applied here. So for our brain, we can know, serotonin, dopamine, acetylcholine, they control variety of the functions, right? None of them was in this organism. So they invented completely new language and new nervous system. But second, when you started to look at these guys more carefully, then it was realized that these guys invented not only a nervous system independently, they also invented digestive system, muscles, many aspects of immunity, organs. So basically, it is a sister lineage of rest of the animals, and I call them aliens of the sea because if aliens will landed eventually on the earth, you would assume they should have brain to make us this ship. But you would never think it will have dopamine and serotonin on the same synapses like in our brain. And these guys exactly like this type of animals.
There's a picture of the
Yeah, so look at this.
microscope.
Yeah, and this is the elementary brain. This is the conductive pathway. No similarity to whatever you'll see in books. And you may say, why is it important? Why is it important to look at this alternative design, right? And I would say, if you know something, you can make something. And I'm working in the Department of Neuroscience, and many of my colleagues study neurodegenerative diseases. In many cases, it's one-way tickets, just simply one-way tickets. You can, if you're lucky, you can diagnose it earlier, you can just slow it down, but never reverse. And if you have one path to reverse it, and if it's not work, it just end. And these creatures can regenerate elementary brain in 3 days. They can heal wounds in 2.5 hours and no scars left. So they already made alternative way how to repair. If you want to go from here to California, you can use train, or in the past you use train, but if you rush, you can use airplane, or you have fun, you can use boat. If you have juice, you can take a bicycle. But it is still different way to do it. So actually, beauty of biodiversity, it's not only just to keep in planetary ecosystem cells. It's combination of solutions to achieve more or less the same tasks. It's a wisdom of nature, and it's our heritage. 3.5 billion years nature did experiment, all bad experiments was eliminated. And you can see huge adaptive space, how to resolve many challenges. And if you're thinking about that some of these animals become dying right now because our stupidity, I think you should at least have a molecular blueprint for that. But I'm convinced, maybe it's my personal opinion, but eventually I think it's most of the people would do this way. There is no way to treat such important disorders, diseases like cancer, Alzheimer, age-related memory loss, without understanding this comparative stuff, simply because we cannot do experiment on our brains. We have to look at the wisdom, and we have to understand how these different Lego pieces can be combined. So we may know alphabet, but we should know syntax, how to combine it. So you can still have Latin alphabet, but different type of syntaxes. But what we even realize, there is more than one alphabet. So to know something is to make something. This is what I think it would be important as well.
Oh, I love that quote when you, in your collaboration with Dr. Kandel, he said, I thought it was going to be complicated, but I didn't think it was going to be this complicated, specifically how the neuron works.
And I would say just another thing, what we start to realize, I mean, just we are talking about scientists. We start to get idea what we do not know. And this is quite striking parallels between study of universe and study of genomes and biodiversity. In space exploration, we realize that matter, what we can see, touch, it's about 4%. Rest of them, dark matter, dark energy. If you look at our genome, approximately 4-5% of that code specific proteins which may have idea about functions, but the rest is regulatory. So it's the dark matter of the genome, more than 90%. And if you eventually look at the biodiversity, as I told you now before, we still have more or less the same relationships. So at least we know what to be done. And that can be done with technology. So 10 years ago, we think, oh, genomic will save the world. But genome is static. You need to see in actions. And what I just tell you, it's just obviously not secret. But even 6 months ago and 1 year ago, I would not imagine that instead of doing one genome, which first genome took a few years and $3 billion, second genome may be close to $100 million, third genome was about $1 million, etc. But it's still static. Today, it is possible to analyze 100,000 cells individually, and not only just how genome works in these cells, what would be produced RNA, but also what kind of proteins they produce, what kind of lipids they produce, what kind of metabolites. 100,000 cells in once, and that can be preserved for a year. This is basically, I think, when you can do this marriage between advanced technology and biodiversity and exploration, I'm really confident that we can grab this wisdom, heritage of humankind before it will disappear. Some people may disagree with me, but I think beauty and importance of some of these organisms comparable, even greater than Mona Lisa. No offense, I do love Mona Lisa. It should be preserved for century and millennia, whatever it is. But thinking about that we have this kind of experiments performed for us, it's a gift to humankind, and if you can lose it forever, I think it's worth 3,000 or 4,000 or 5,000 euros per species to keep it
Leonid,
for generations.
Our last voyage that we're talking about is a voyage we're taking in the future this spring. We're heading from Maine up to the west coast of Greenland on a fantastic vessel called Arctic Earth with David Conover and his crew. Why Greenland? Why Greenland now? You've called this voyage to go up there a voyage through time. What did you mean?
Why Greenland? Why not?
Oh, by the way, this is a new species we found last year in British Columbia. We call it the Farah fossitophore. It's not official, but you get that. That may date me.
Right.
This is, by the way, cilia off of the ctenophore. Interestingly, it creates no perturbation in the water.
Basically, all of these animals, like stealth predators, they can approach.
These are also larval organisms, talking about biodiversity. This I just put in there because it's super cool. I mean, there's a lot of science to it, don't get me wrong. And this is a lovely organism called a sea angel.
So, and it eats sea devils. So, goal is to sequence angels and devils, and just to see which genome is better.
These organisms may have clues to mood regulation.
But
I think by just watching it.
What read about angels? This is called theropod mollusk, and they're very sensitive to environment, to acidifications. Very good reporters, like tipping points.
These are diatoms. These are responsible for a huge amount of the carbon capture in the ocean. And these are mollusk larvae.
Yeah.
It's pretty incredible, huh?
Yeah.
All right, so going to Greenland.
Well, as I said, why not? And I think everybody in right mind, if has the opportunity to go to new place, will go. So
we're gonna sail up there.
But
I just wanted to show you this vessel because these boats are really built to go anywhere in the world. These are all aluminum construction, incredibly durable. And we're going to head up in the ice, and this is the previous trip.
But I can tell you why. In addition to fun and probably great opportunity to discover a lot of new stuff, because survey for many, many species over there was done systematically, sometimes 100 years ago. And there are some organism, if you don't know, which lives in that area, may live for a few hundred years. So they have enormous lifespan, longevity. And some invertebrates in polar area may live 4,000 years, 10,000 years. You may just still ask questions how and why. So in addition to that, and there's many, many other solutions. But what is important about Greenland and many places like that, two things. First, it is a tipping point, like pulse, one of the pulse points for our planet, for multiple reasons. One of them, it was even published paper 1.5 years ago in Nature Communications. They said, well, let's say if a liquid melting will be faster than its current, Gulf Stream can be ceased to exist in interval from 2025 to 2090. So no wine in France. But there is another aspect. I told you about wisdom of nature. Origin of animals started after global glaciations. So life and animal ancestors in particular were exposed to this kind of catastrophic changeable environment several times. They accumulated variety of the strategies across different species. And in some way, this is travel to the past, because if you're now past and we're now present, we can predict a little bit of our future. So in some way, it's a win-win-win situation. You can go to the baseline, which has happened right now, just you can preserve species, because some of them can be genetically preserved. You look at the past wisdom of life, adaptation to stressful condition of changeable Earth, and you probably, if you can do with advanced technology, you probably can develop models. And I tell you exactly what we don't know. We have, let's say for animals, 33 phyla. It's only 5 grand designs. And out of the 33 phyla and 5 grand designs, we only have information about 1 grand design and only 5 phyla. So it's like in periodic table, we only have one knowledge about one period. So you'll not do any kind of synthetic chemistry. So that is basically, I would say.
This slide here is from about 600 plus million years ago. Some of the changes in the melting that are happening in Greenland are replicating some of the catastrophic changes at this time, which is really when we think, or we, Dr. Moyes and other biologists believe that's when the changes were so dramatic they really forced the creation of complex animal life. These are the tipping points. We talked about them. There are These are the tipping points that we talked about, and there are a number of them around the world. And one of the things when we talk about biodiversity and biodiversity baselining is getting out to these areas and documenting them as quickly as possible. And Greenland is certainly one of them. So, Leonid, I want to move on to the boat again and why we're going with a sailboat again, and how you feel that these mid-sized vessels are good at this stage of investigating biodiversity of the planet and applying this technology.
Well, it's cost efficient, number one. Rather just to send a big mothership with cost millions of dollars, you have to do exploration. But what I would like to say, it's always analogy to Star Trek. If you have advanced technology, so it's better to go with versatile mobile vessels, do initial mapping before bring heavy cavalry or big boats. And because we have about 10 tipping points around our planet, so I think it's cost efficient for humankind to put advanced technology, link it to explorations in critical points and keep a baseline for quite a while. So did I answer your question?
Yeah, that's great.
Yeah. Cheaper, faster, more efficient, and no argument, right? So only you and I potentially can argue.
Final thought. If you look at, in your career of exploring the oceans and thinking about the Challenger and Beagle and other vessels, would you say that we're at the end of the beginning? Where you think we're at the beginning of the end of ocean exploration? Where in that arc are we today, in your opinion?
Two aspects of answering to your question. First, people may realize, if you read the literature, majority of exploration was done people on the boats, and it was only from 1 to 5 scientists. So it was a great Challenger expedition, which really created modern oceanography. How many scientists was there for 5 years? 5 scientists. They create entire field. Darwin was one on the Beagle ship. So I think it's one of this advantage. But then Peter quote Winston Churchill, I guess, right? So Churchill said, it's not the end, well, correct me if I'm wrong, it's not even beginning of the end, it's end of beginning. But I think I should say it is beginning of the beginning, because we have technology and they will be just better developed. And we now at least 4% or 10%. So I think correctly to say it's the beginning of the beginning and have this spirit of exploration, which will help us to live much longer, get rid of cancer, because many marine organisms don't have cancers. Many of them immortal. Some of them regenerate brain, keep memory as long as we are. So why not? Plus side effect to save our planet and save our home. So win-win situation. So I think
Maybe
it's
so.
beginning of the beginning.
Maybe so.
And I'm glad that we're in an Explorer Club. So yeah, so why don't we start the NOAA and retrace great explorers such as Darwin, Challenger.
That's our plan. So we'd like to open up to questions. Yes.
Please wait for the mic. Sorry.
Yeah, there's a.
I'm so sorry. I didn't want to ask a question, but my God, this is just. So I work for the I study global environment facility projects around the world. And I work with UNEP and UNDP on biodiversity, and I'm involved in that whole policy stuff. You are mapping baseline of biodiversity, like at the lowest form. And so I'm wondering, like...
Functional biodiversity.
Yeah, functional baseline, which is really the bottom of that tree of life, right? The single in the oceans. So I'm wondering, like, how come this work at the bottom, the root of the tree of life, has not been recognized politically in these global biodiversity forums, you know, that and these political things that are happening globally, and why is this not seen as a baseline for all of the work that we do on monitoring global environment work?
Should I answer it? Yeah. Extremely good questions, and usually there's a two answer to your questions because science. Complicated. Why now? Why not yet recognized? That technology, for example, which I mentioned to you, which was cost efficient, became available 6 months ago, right? And before that, you can do single cells, but it was still more expensive. So you can do genomic, but you cannot really scale up. And technology was, I put it, not very democratic. So democratic means that you can move it, share it very, very quickly. So it's number one. Actually, 3 answers to your questions. Number 2, because technology and marriage technology with exploration happened recently, psychological barriers, it's a major, I would say, in my mind, it's not like technology or making the ship sick or whatever. It's just a way to convince that it will work. Usually in humankind it happens, you have to do that first, and then everybody will do it. Some technology which is currently today implemented in environmental sciences were development 10, 20 years ago, and it's only recently started to apply. So I think this would be important to work together for scientists, because sometimes they live in their own universe, and public, and deliver this knowledge and opportunity that we can do it as soon as we can. So more publicity would be done. And in this case, because people who involved in many strategic political decisions, they would need to have at least not one voice, they should have a variety of voice and success. And in this direction, I would say there is also critical mass. Let's say you cannot be half pregnant, right? So you either pregnant or not, maybe not a good example, but anyway. But normally what you need to do, you basically need to have, like Star Trek, you need to have critical mass of equipment and do it. And I would say Peter and I and many other scientists, we did incremental, step by step. We validated one and other technologies working. But right now it seems to be all of this come together. And bottleneck for that, real bottleneck for that, is to get access to oceanic vessels. when you can really prove it. And with all supports scientists have from variety of the grant agency, sometimes funding both operation, especially if you're a neuroscientist or clinical scientist, is challenging. Yeah. And this is what required to get critical mass, change psychology. And for me, we usually live in our worlds and we try to push limits. But reality, when advances in science go to public, in my opinion, sometimes it's 5 years or 10 years, etc. And this has hurt us from all angles. So I think we should work together and try to find a way to work together. That is.
Yeah. Yes.
I have a question. This is a little open-ended, but I mean, and obviously incredible the work that you guys are doing. But this is a bit of an open-ended, I guess, on some level philosophical question. But you know, something you alluded to, I think, throughout this lecture is the notion that sort of we are in a race against time. You know, in terms of these species.
Race to say species.
Right, but you know, I think there's this tension nowadays, right? Which is, on the one hand, technological progress is incredible and remarkable and getting better. At the same time, the damage to the planet, sort of the ecological problems, you know, keep compounding. And so, sort of, as someone who's at the forefront and sort of you see the damage, you see what's going on, things that are changing, but you also use and see the technology. Are you optimistic? Do you think we're going to be able to, you know, are we going to win the race against time? And sort of, kind of your thoughts on that, on where things are going, and sort of that tension between, you know, the clock is ticking, but at the same time we're figuring a lot of stuff out.
If I would not be optimistic, I would not sit here, just to begin with. Then I think, well, how to say? It's not my saying, but Edison said. Pioneers recognized by number of arrows in their back. But sometimes you really have to be first. And in science, if you survive, you basically, if most of the people say no, that means it's just right way to do. And you may, I never take projects which have more than 30% of success. So, but if you have 2 or 3 projects, one of them would be successful. And I can see it. Why not? If you can just do it, preserve cells, do analysis, why not to be first? So, and I'm pretty sure that many scientists think as I am. So I'm super optimistic. And I would say we do losing a little bit time, but we not lost a lot of time. Because what is required is you reach critical mass and then it will go up. So question is, we can reach it in 2 years or 3 years, but definitely we'll reach it in 5 years. Because even right now, everybody talking about biodiversity, everybody talking about importance of baseline monitoring. But biggest problem, no, sorry, wrong word, challenge. We have no problems, we have challenge, we have to find way how to overcome them. So biggest challenge, I think, for us and for many scientists to understand and realize we will not be able to fix big, important, critical disorders like cancer, mental retardations without our comparative evolutionary stuff. Cancer is disease of genome. And genome, it is accumulated knowledge, wisdom of many millions of generations. We do have Neanderthals DNA in most of us, I hope. We do have diversity, a lot of, well, good example, one of the genes which control our mood came from bacteria, right? It's involved in metabolism of serotonin, right? Good sleep, good mood, bad sleep, right? So our genome is more than, and we will not understand future without understanding the past. And many people will thinking about, oh, if you invest $10 billion in cancer, it will be solved. Richard Nixon's, when war in Vietnam was finished, right? He said, oh, let's do war against cancer.
A long time ago.
long time ago, right? And we're still not there because we realize we need to understand how many pieces work together, what would be language, syntax of life. And that requires dynamic. And this only can be done in this broad comparative aspect. In evolutional scale, across majority of the lineages, an ecological scale, when we have a resistance or resilience to anthropomorphic, anthropogenic factors, and then we have to do experiment. So I'm optimistic, super optimistic.
There's also a future aspect to it because we're hoping if it's open source, it's not just for scientists who are working today, but once it's digitized, there'll be scientists in 20 years, 40 years, 100 years who may be able to find secrets that we can't comprehend.
I will have proactive suggestions to your question. Proactive suggestion is...
Let's get a couple more questions. Okay, Ian,
I'll give you an even better answer.
Hi. So this is obviously a huge, awesome, grand project you guys are doing. But I guess in one respect, you know, if someone were to come here and say, you know, my project is I'm going to map all of the land creatures, that would be, you know, a very big project. And you guys are, from my understanding, trying to do, you know, map all of the animals in the sea or all of the organisms. What is strategically the crux for you guys or the bottleneck right now? You mentioned the oceanic vessels, but how do you think about, you know, vessel development versus the neurobio side of things versus scaling the project? What does that look like for you?
Perfect question.
Really good question. Let me give a layperson's answer to that. We can actually take representative samples of a number of the different lineages, which will give us basic clues to the architecture of that lineage without sequencing every species in that lineage. So it's way too many. That's a great question. There are a lot of organisms out there. So it's going to be strategic to, especially phyla that have not been well studied, Leonid.
Okay. So first let's understand why sea, not land, correct? Yes. All right. Very good question. So diversity of life in our ocean much greater conceptually than diversity of life on the land. With all due respect, I love right. So land ecosystem must be preserved, should be preserved, but it's common knowledge. Everybody will do it eventually. For ocean, even if it's 71% of our planet, it's much more challenging. So as I said, I pick up only projects which have less than 30% chance of success. So that is one of them. So ocean is more complicated, but I think it's more needed because of greater diversity. It is control health of entire planets, 71%. And if land diversity, well, it's recognized, it's still out of unknown. But understanding about diversity and operation in ocean, especially in deep ocean, it very, very, very superficial. So I think we should do it. Yeah.
To your question about how we strategize, I have to say that after every voyage, we are reassessing that because we are seeing almost always, sometimes you said 200 to 300% more than we expected to see. And that changes the gauging of where we're going to go.
I can give you a number, which is also important to have number, especially in New York, in terms of money. So we have approximately 240,000 described species, marine animals. They belong to just 30 plus phyla. To get all of them as a blueprint, which will be 95% of all wisdom, right? You know how much cost it? $250 million. If you're thinking about representative lineages, there is a taxonomy, there is a 33 phyla, there is 100 classes. Let's say just do very simple math. So for 10 millions, you'll get 80% of wisdom of. So is it doable or not? I think it is. And on Earth, on land, we have like number of the phyla, but it's much less. Life originated in ocean. Ocean is the only hope as a buffer of entire planet. It's linked microbiota, bacterial world with us, all carbon, nitrogen, etc., etc. It's the only way to do it. Then sooner we realize it, then better. And I would not against any kind of space exploration because it drives technology. But at the same time, we do see aliens in our sea, which is cheaper to get it.
We actually have a slide of Men in Black that we can use tonight.
Yeah.
Yes. Felix, please choose. Yeah, thanks.
Thanks. So there's this RNA world hypothesis that I'm sure you're familiar with, that RNA sort of predated DNA as the original genetic information. And so I was curious if you think, one, RNA life forms still exist, single cell RNA life forms, and two, would you be able to capture those using your
Yes,
technique?
it's so easy to answer your question. You are one of the example. You may think, and I don't want to capture you actually, but indeed, RNA molecule is very versatile. It not only can store information, it can do enzymatic reactions and can be structured away, and that was RNA world. You think it disappeared? No. We calculated how many different kind of RNA exist in one neuron of this C-slug, aplysia, which Eric Kandel used for study of learning and memory. You know how many different type of RNA in a single neuron of C-slug? 120,000. And only a small fraction of them encode protein. Rest of the regulatory. So RNA world exists in every cell of you, hopefully me too. But critical way is how they interact all of this together. But there is this tenophores, just to explain the questions, there is some regulatory mechanism related to RNA which is called microRNA. These do not exist in the tenophores, but they have some another alternative way how RNA can be used or reused for alternative functions. And the beauty of this, I so love your questions, I cannot stop. Reason for that, because RNA perform multiple functions, but it's nucleic acid. You can synthesize it. You can make probes. And people already do it, scientists already do it. They make versatile probes to detect cancer cells, to control cancer cells, to eliminate it. And I'm pretty sure this technology can be incorporated in many other ways. So animals, nature, already provide variety of the way how to do this combinatorics. Again, numbers is critical. Total number of particles in observable universe, proton, neutrons, neutrinos, and so on, approximately 10 to the power 92, 93, 94, something like that. If you will think about 400 cells, neurons, in a leech ganglion, and you can assume existing knowledge about that, number of random combinations of output would be 10 to the power 1,200. So it's impossible be smart scientists to switch on this and this and this and try to control it. What more likely, better to screen it and find RNA and many other combinations. So RNA worlds.
I guess the question is whether or not there's something that has RNA but no DNA.
Well, bacteria, many prokaryotes, yeah, viruses. So DNA is good invention because it was released for enzymatic functions and just keep storage information. Viruses, they obviously like RNA, but I don't know any example when eukaryotic cells may have only RNA functions, but RNA world exists. All of these communications, and Eric Kandel, in fact, in his research, good question, which I still try to answer. Why do you remember your first kiss? So thinking about you form memories, and then 5 years after that, you still remember it. Somebody, if you like, you can remember it in 60 years or 70. But half-life time of most of the proteins, just let's say about 2 weeks or less. Lifetime of metabolites can be even less, right? So how can you keep this kind of long-term memory? And two ideas, which is also because of marine sciences, end up with, well, first, DNA stable molecule, you write down this information in the DNA, the one idea. But how it's done not only protein, but then RNA. And second, what is exists in our neurons is, Eric Kandel called it like a dialogue between contact between cells, synapses, and nucleus. And critical mass of these are involved in a variety of non-coding regions of RNA. So RNA world exists, multiple perturbations, and this is the reason I will say you cannot resolve cancer or longevity or memory problems without comparative analysis. I mean, just one way to do it. It's only one way, no alternative so far.
So I had a question about the capacities that you're building on the vessels, especially that you're planning, but including ones that you currently have out. Obviously, you have a large focus on microorganisms, and I was curious to hear if you've also prepared protocols for collection on other vertebrate species, and obviously there's great opportunity for opportunistic collection.
Great. Okay.
So if you could speak to that.
Yeah. So number one, regarding to vertebrates, and in some way it was initiated in New York. There's already ongoing program basically to sequence all vertebrates. And good things about it, there's a very few species of that. So like mammals, about 5,000-6,000. I mean, just there's a very limited number of the species of vertebrates, which is doable, important, and people do it all of the time. Then second part of your question, microorganism, right? So these are not microorganisms. Some ctenophores may be up to 1 meter. So what is the most critical, why we're looking on this, because there's a bottleneck. Let's say climate changes. It happens so rapidly, we call it 6 extinctions, right? And it's because we destroy habitats and invasive species. The only way for life and animals to survive is escape. And how it's happened, most of the organisms developed 2 stages of life cycles, biphasic life types. You have benthic organism and you have larva, which is a swimming, pelagic, planktonic. So many of these creatures, planktonic, it is basically babies, which looking for place to live, to settle, basically dispersal, settlement, metamorphosis. So this is like hourglass, bottleneck. If you really understand that, and actually this is even more interesting. So you think just too big project never be done. But you know how many cells make this life decision, a single decision? Every time in the ocean right now, billions of creatures making single decision, basically swim or settle down. And you know, this system has approximately also 5 grand design, 50 minor design, but it's mediated by from 10 to 50 cells. See, small number, decision-making circuits from 10 to 50. And then it's lifetime single decision, most important, swim or die, eat or not to eat. Very simple. So I would not say just we only study microscopic. We try to strategically pick up bottleneck and less explored part. And of course, adult animals as important. Like these animals which live 1,000 years in Greenland or like 10,000, 4,000 years in Antarctica, why not? They're big, they can be a couple of meters.
The other thing is that as we expand our capacity, we have been bringing on more and more scientists who have different scientific goals and outcomes. But the idea is to make the boats modular so that we can bring equipment on and off very quickly, so to reset it for different scientific goals. Yeah.
I think we can do one more quick question here.
We can do more questions if you're interested. You know, some people stay. I think the more connections.
Hi, thank you so much. That was just like wonderful, incredibly informative. I sort of have a 2-part question. So you mentioned that you're doing research specifically in these biodiversity tipping points. Do you have a methodology for how you choose exactly where to sample within the areas that you're looking at? And I was also curious if you've done any tracking of changes in biodiversity over time with the areas that you've researched.
First, strategic points. Yes, Greenland, one of them. Antarctica, it's another one. There's a few spots, critical spots, let's say in coral triangles, etc. So at this moment, I think 3 or 4 points in polar regions and tropical regions, it's enough. Because what I'm really feeling is, it's important to not only validate, but also proliferate technology. It's like if government will make a good highway, people will run it in all directions. So I think major challenges, pick up 1 or 2 points, keep baseline, and set up platform which will be open for virtually everybody, including science citizens, to be used. So, and I would anticipate, you can tell me predictions is always wrong in science because about 1.5 year technology adjusted. But I'm thinking that capacity of the technology, if you have platform, if you have this pathway, would be sufficient virtually. I told you $250 million to grab majority of animal species and planet on oceans. That can be a quarter of that, more likely. So why not? And in my mind, what is important is there is out of 33 files, 20 completely untouched. So that would be probably more important. Keep baseline in one place, but try to grab blueprint, or maybe this decision-making circuits first, and just to see how it's operating, and then, well, you look on the rock and you probably find another one. You can go to the mountain, and eventually, after this pass, you can see, wow, this is entire perspective. This is what I think we should do eventually. It will happen probably in 2-3 years.
This is part of the beginning problem. So to your question, there's really not much baselining at all at this level in these areas. So our strategy is basically we plankton haul twice a day, and almost everywhere we get interesting things. We have been consistently surprised almost everywhere we went. So we will hone it in. Don't get me wrong, there has to be a strategy. It's a huge ocean, but we're very much at the beginning of this, and the methodology and the techniques and the data analysis. I'll give you an example. Lena, you were talking about this summer. So using AI, when you do metabolomics of one single cell, it used to take something like 2 months or a long period of time for the programs to work and do the patterning. And now I think it's down to 5 minutes.
Yeah, something like that, even
right?
faster.
And so a lot of this is we just have to get these, but the biggest bottleneck, as Lena was mentioning, is we do not have enough of the sushi grade samples yet. That's it. So we have to get out there and get them. Can I?
Hi, I'm actually from the Vertebrate Genome Project, along with several of my colleagues.
Exactly, perfect.
I would say there's a few more than a few thousand vertebrates, but that's besides the
20,000, how many of them?
70,000.
70, well, okay, still smaller fractions than 2 millions in the sea.
It is a lot,
But we're not counting.
it's fine. I would just like to know what sequencing methodology you're using and the quality of the genomes that you're getting.
Well, in many cases, we don't need to have a chromosome level assembly. But usually, there is PacBio long read type of sequencing or nanopore sequencing, which help us to do assembly. And of course, it's complemented by Illumina or Element. This is what we try to do. Yeah, so I think we try to be open to everything. So there is a reference genome. When against these reference genomes, we can map close related species. So for reference genome, should be done for all these 20 files which left unattended. And for draft genomes, yeah, I think it can be close to 100 species. But maybe what we should do if you invertebrate project, maybe we should combine efforts, because even with all importance, let's say wolf, fox, and panthera and lion, etc., they're still quite similar. And if you look at the spectrum of diversities, in vertebrates, it's greater. So I think both projects are extremely important. Vertebrates closer to us, but I think both ways should be done, since vertebrates is easy to sell. In vertebrates, it's more complicated to sell.
Felix, yeah.
I'm sure you both will stick around and feel free to go up and ask any lingering questions. But I think we should all give Peter and Dr. Moroz a round of applause. Thank you so much.
And finally, a huge round of thanks to the Explorers Club for hosting us and the ability to share our work. Thank you.
That was great. Oh, yeah, it was great.
If you're just doing this,
First time.
Thank you so much. Is extremely important. Yeah, it's not always the best way.
Thanks very much. Fascinating.
You're welcome.
Bye.
Hey!
Yeah, it's actually 10 blocks away.